This video is a short 5-minute version of a longer presentation that I'm working on about the SPDI Data Model. One of the projects I've been working on is creating a database to track variants that we encounter in next-generation sequencing. And the challenge that we face with aggregating information about variants is that we typically identify them using HTVS nomenclature or genomic coordinates, and that makes it complicated to update our bioinformatics pipeline and still be able to access that aggregated information. If we want to change transcripts or reference genomes, we need to be able to find information about that same variant in its new context. And since NCBI manages several large databases of variants, I decided to do some literature searching on how they manage this problem, and that's when I came across this paper about the SPDI data model that they use. So to get right into the SPDI data model, it stands for sequence, position, here we go, yep. Sequence, position, deletion, and insertion, and it provides a standardized representation of genetic variants in those 4 components. The reference sequence where the variants are found, its position, and the sequence that's deleted, and then what's inserted. So I have an example here. I'm just going to go through this really quickly. I'll go through this Jupyter notebook file in more detail in the longer version of this, but here you can see this variant represented in VCF format. This is what it would look like in HGVS, and then for the SPDI, this is like the genomic coordinates, the position, and then the C is replaced with the T, and that's for a single nucleotide variant, which makes it a little bit easier. But you can do this using, genomic, transcript, or protein reference sequences. But why is it significant to use the SPDI data model? Because it offers a consistent and clear way to represent variants. Additionally, it effortlessly converts variants between popular formats, such as HGVS and VCF. And for tools like dbSNP and ClinVar, SPDI, which is pretty valuable, which I'm going to demonstrate in a little bit. But importantly for developers, SPDI is easily machine parsable. Now, SPDI provides a consistent language. It still has the challenge of low complexity repetitive sequences, and this is where the variant over precision correction algorithm comes in. It ensures that equivalent variants are framed in the same way, and its process is, I've got a graphic here, and again I'll go into it in more detail in the longer version, but for the quick version, it just ensures that all equivalent variants are framed in the same way by reducing the variant to its smallest form and then expanding it within the repeat region, giving a unique normalized representation termed the contextual allele. And then when you have all the contextual alleles for the same variant using different transcripts and reference genomes, etc., then you choose one that's the canonical allele that is meant to represent all of the contextual alleles. And to sort of help with this, NCBI offers this variation services. It's an API that you can use to work with the SPDI format, HGVS, RefSNP. And so I have an example here. So keep in mind that this is an unfinished presentation, but here I have an example VCF with a few variants from, I think, BRCA2 gene. And especially take a look at this deletion here. This is in a repetitive region, so I'll show you what it looks like after the variant over precision correction algorithm is applied. So I have a little script here that will parse through this VCF, use the API to get it'll parse the VCF, and then it will send that VCF information in a API request to the variation services, which will return the SPDI format, which then we will use to get a bunch of links to other information that variation services has on that variant. We're going to generate the contextual allele, then we'll send that contextual allele off, and we'll get a bunch of other links. So Let's parse through that. You can see here is the contextual allele for each of those variants in the VCF file. And this is the variant over precision correction algorithm applied one for that T deletion that's right here. And you can see it expands it to the entire region so that in the future, if you were to store this as a field for that variant and you wanted to add some annotations to it in your database, in the future, if it calls that T deletion at a different position in this repeat region, after applying the variant over precision correction algorithm, it will normalize that so that you'll always be able to find the previous times that you did that. And by applying this along with the SPDI format, NCBI was able to reduce redundancy in their databases by up to 50%. That's for like dbSNP and ClinVar, and I'm not sure where else they applied it off the top of my head right now. But so we can take this SPDI format. We're just going to work with this last one here from now on, not the other two. We can take the JSON that's returned from this. These links that I have here are just like sending the request, and then you get this JSON formatted return. Then here it gives you all of the different like all equivalent, contextual, canonical, representative, etc. And you can use all of those from the JSON format. You can parse them, and you can get links to you can create other API request links to get things. So and I think the contextual allele, the all equivalent contextual allele link, is particularly useful because it will return an SPDI for every transcript and reference build, so that if you change from HG19 or HG38 or you update transcripts, you can still look up your previous encounters with that variant. So let me just... So I have something that sends an API request using the all equivalent contextual here, and then it parses the response, and it kind of cuts it off because of how Jupyter Notebook look. Well, I guess I could just open it here. And you can see it's like, this is, I think, HG38. This is what it would be for, HG19. This is, I don't know, some other genomic coordinates. And like, this is probably the main transcript. That's a previous version of it. There's some other transcripts here, other NG numbers, but it's everything that it has in the NCBI database for that variant is there. And then you can use the canonical transcript that you get back from that, which, we have it up here. So here's the canonical representative one. And so we can encode that in API request to ClinVar, and we get back the ClinVar ID, and then we can use that ClinVar ID to, we could create link outs to the ClinVar page for it. Okay. But then we can also use it to send a API request to ClinVar for information about that, and you get back this XML file. You can parse the XML file for whatever relevant information that you want on that variant. So here we have like the name, protein change. This is HGVS format for Indel. When it was last evaluated, submissions, clinical significance is pathogenic. Yeah, I think I was where I last left off when I was working on this. I was trying to set up Genomad links, but it doesn't work exactly right with SPDI, but I know that they do want, like if you're doing an indel, they do want to know adjacent, like what the reference is. So you can't just have a dash there, or the alt. You just can't have a dash for indels. So it's good to have like the context. I just haven't figured out how to parse it for this yet. But for like dbSNP, yeah, dbSNP, it would also use the SPDI format. So yeah, I think that incorporating the SPDI data format into any variant aggregation, application or annotation pipeline could be really useful for long-term use and flexibility. Thanks for listening, and if you're interested in a longer video, when it's ready, let me know. Teams or email, and I can send it to you, and I will I'm going to post this, what's it called, Jupyter Notebook file to a GitHub if you wanted to take a look at it. Oh, I guess this ended up being a 10-minute presentation. Never mind, 10-minute presentation instead of 5 minutes. Anyway, thanks a lot. Take care.